Okay, I'd like to present my work. Hello, everyone. I am Pil Kim, a professor in the Catholic University from South Korea and leader of a research project developing novel food ingredients for alternative proteins. Also, the former CEO of Hemolab Company. Today, I'm going to tell you a story how we found a high vital microorganism for manufacturing a single-cell protein ingredient and how we achieved an unexpected result that is relevant to gut health and slim body to animals. Single-cell protein refers to biomass or protein extract from cultures of edible single cells. Compared to animal, plant, insect proteins are from breeding or plantation processes, which belong to agriculture, livestock farming, aquaculture, fishing, and it requires large land or sea. Single-cell protein, however, from fermentation process, which belong to manufacture, and it happens in-house, so not much field is required. Single-cell protein have many merits over conventional proteins because it can be manufactured in-house, which means less field, less deforestation, less greenhouse gases, less water usage are required. Second, it could be a good protein food because it has high protein contents you can easily understand at the animated cellular components. Most of the intracellular materials are composed of various proteins. Third, it could provide additional health or taste encouraging functions. People empirically know that food ingredients such as chlorella powder and yeast extract are health beneficial. Both are derived from cultures of single cells. Now, let's talk about single cell protein story from a high vital cell. At first, we wanted to find a bacterial strain made up of a well-balanced mixture of many vital proteins. One might say like vital proteins are proteins roles in essential to life process, such as life energy generation process, detoxification process, cellular redox balancing process, and the many signaling process. The vital coenzyme part bound to protein part could be heme, quinones, flavins, iron sulfur cluster, sulfur-containing amino acids, and many minerals. To find a high vital bacterial cell, we initially hypothesized that if we build the laboratory conditions having gradually increased flow in continuous culture, only high vital cells could be eventually dominant in the culture vessel, which resembles a natural evolution process. Indeed, natural microbiota initially having various strains were gradually washed out and fewer strains remained in a couple of months. The remained strains were capable of rapid growth, so only high vital strains were enriched while low vital cells had gradually removed. Among the remained strains, we isolated two colonies, those showing the reddish color because it was the easiest way to select a bacterial strain harboring heme protein, one of the most useful molecules in the life energy generating vital process. When two kinds of protein ingredient were manufactured in a pilot plant, they also showed a reddish color and smelled like a dried jerky. They didn't show any toxicity from mouse test and based on the single dose and the repeated dose. So we thought this ingredient would be good protein source for animal nutrition, especially for iron supplementation, considering heme's red color. Now, and we tried a field test. The one of two ingredients were tested to see if it is be suitable for the weight gaining nutrition for broiler chicken, the chicken for meat. The conventional bird diet were mixed with a small portion of new ingredient and fed them for 32 days to chicks, expecting more meat production. But the result gave us disappointment because the supplementation did not help weight gain. Here, even though feed efficiency was higher than control, amount of the consumed feed were lowered. After repeat field test, we concluded this ingredient was maybe nutritious considering the higher feed efficiency, but chicks did not like to eat much, maybe because of its smell or taste. So we decided it is better to apply this ingredient to dog food that likes meat-smelling ingredient. Anyway, we collected the remained organ after meat sales and counted the numbers of lactic acid bacteria in animal cecum, thus not used for meat sale. We found that more lactic acid bacteria reside in the cecums from the ingredient supplemented group. We did not know why, and this was the first clue that the new protein ingredient might enrich lactic acid bacteria in gut. To verify the clue, the protein ingredient was supplemented. A dark and light blue portion indicate the Firmicutes phylum, where many lactic acid bacteria belong to. Surprisingly, live merchandise feeding group resulted a decrease of Firmicutes instead of increase of proteobacteria such as E. coli. Later on, we figured out that the probiotic product contained sugars for its sweet taste, and it must have caused the proliferation of proteobacteria, which can grow a lot faster than other anaerobic bacteria in sugar-rich condition, resulted the decrease of Firmicutes portion. However, the ingredient somehow suppressed the proteobacteria, enhanced the Firmicutes portion along with the enhanced bacterial diversity. This result implied this ingredient has potential to control the imbalance of gut microbiota. In another field test, the protein ingredient was used for as part of medium component for cultures of commercial probiotic strain. Bifidobacterium cells grown on the ingredient harboring medium got reddish, and they survived more than control after the manufacturing process. This result also support that this new protein ingredient has potential to extend shelf life of the probiotic product. For your information, the single-cell protein ingredient initially developed as feed was not allowed for probiotics manufacturing process because it was not a registered food ingredient at that time. The proliferation effects of the protein ingredient were not only applied to the commercial lactic acid bacteria culture, but also to other representative oxygen vulnerable obligate anaerobic bacterial cultures, including microbiome drug candidate strains. Therefore, we were able to imagine this new protein ingredient might contribute to balance the gut microbiota dysbiosis provoked by the leaked oxygen from a blood vessel. Anyway, the company decided to apply this ingredient to dog food component instead because it smells like a dried meat. We manufactured a meat alternative dog food product for sale. Because of the jerky smell, every dog likes to eat meat alternative product very much, being happy dogs. It is actually in sale, so you can test it to your dogs by yourself. Besides the dog's preference being happy dogs, we collected reviews from customers. We also asked some customers to return the dog's feces before and after the product feeding. Many customers reported symptoms related to dogs' gut health as well as being happy dogs. The reviews were like a better stool consistency, less put-lid smelling stool, less diarrhea, less inflammation, and so on. When we analyzed the fecal microbiome before and after the product feeding, we found the increase of Firmicutes and the increase of diversity were the same trend as the previous mouse test. The individual gut health relevant bacterial change include increase of fat degradation, more immune lessons, and so on. You can find the detailed result in this publication. All these results imply that the high vital protein ingredient resulted the dogs get healthier. You can 2 years before this study, we had preliminary tested the controlled feeding of the direct ingredient to a few dogs. We had found a similar fecal microbiome trends relevant to gut health. You can find detail in this publication. One thing we realized later was the slight lower body weights of the tested dogs. It looked like dogs get slimmer by the ingredient feeding, though it was not statistically obvious, but it reminded us the slower weight gain case in the chicken test. To verify this symptom, we designed an obese mouse experiment induced by high-fat diet with protein ingredient supplementation. As you can see, the shapes of mouse, the high-fat diet fed mouse turned to big roundish shape compared with normal calorie diet fed one. The ingredient supplemented group, these two, however, looked still bigger than control mouse, but more angular and slimmer. In spite of distinguishable body shape, the body weight were in a similar range. The fecal microbiome analysis told us the similar trends with the chickens and dogs once again being healthier gut. When we look at the body weight variation, the protein ingredient fat group showed a slight less weight, but not significant. When we measure the dissected fat tissue weight, the subcutaneous fat weight was significantly lower, while the thigh muscle tissue weight was significantly higher. This data agreed with the distinguishable body shapes. When the blood was analyzed, we found the protein ingredient supplemented group showed the lower triglyceride concentration and lower total cholesterol concentration with statistically significant, explaining lowered fat tissue. Also, C-reactive protein concentration, the biomarker of inflammation, was lowered by the protein ingredient supplementation. You can find this data in detail from this publication. Then we were wondering the sequence of actions, how small portion of new protein ingredient had led the slimmer body shape. So we repeated similar experiment with a normal calorie diet or high-fat diet background. After 4 weeks, the ingredient supplementation resulted the same trend result. Those are lower blood triglyceride or total cholesterol, lowering fatty acid metabolism in the liver tissue in both diet background without statistical significance. Histology of the skin fat tissue gave us the distinguishable decrease of subcutaneous fat tissue, once again in both diet backgrounds. Then we hypothesized that these slim body phenomena might have something to do with the gastrointestinal tract, so we detached their intestines. Mucin is known as a slimy surface material covering many animals' organs composed of a heavy polysaccharide on peptide backbone, which is secreted from the special epithelial cell named goblet. And it is known the more mucin in the gut tract represent the healthier host and healthier microbiota. When we photographed the mouse's large intestine, proximal and distal colon tissues showed the tendency of higher goblet cell distribution in the protein ingredient diet group. You can see more blue spots in the bottom pictures. To acquire quantitative data, we measured the mucin backbone protein's messenger RNA concentrations from the gastrointestinal tissues. There was significant increase of mucin in the ingredient fat group across the intestine tissues. Based on this result, we proposed a mode of actions, how the new protein ingredient acts to host animal become healthier and slimmer. Small portion of high vital single-cell protein ingredient would enrich healthy anaerobic gut bacteria in cecum. The promotion of the health beneficial bacteria would signal to stimulate goblet cells to secrete more mucin across gastrointestinal tract, leading a healthy gut status. At the same time, some beneficial bacteria reside in mucin layer, such as Lactobacillus species, upregulate their lipid degrading physiology. This change would lead more lipid utilization by bacteria and lower the lipid availability to host animal. Also, the dense mucin gives a hydrophilic barrier interfering hydrophobic fat components transport into bloodstream. This consequence result lowering the fat tissue accumulation and lower inflammation in the host animal. It is well documented in the pharmaceutics research field that gastrointestinal mucus layer acts as a physical barrier to hydrophobic drug particles. This sequence of actions by the high vital single-cell derived protein ingredient is under consideration, so you can see the detailed data soon enough. My one colleague recently published a paper that a porcine cell, pig cell culture was also promoted by the vital protein ingredient instead of expensive fetal bovine serum components. The promoted porcine cell by the protein ingredient did not express any cancerous signal, and the porcine cell were in more energy generating status, considering the most expressed protein was cytochrome P450. Data from this paper also agreed with the result that the new protein ingredient could stimulate goblet cell in the gastrointestinal tract. This is the last. The single-cell protein ingredient promoted many cultures of obligate anaerobic bacteria, including lactic acid bacteria, and also goblet cells, resulting mucin promotion, and also porcine cell culture for cell-based meat manufacture. The promotions by the ingredient were probably comes from its well-balanced amino acid, including sulfur amino acid or aromatic amino acid composition compared with another alternative protein sources. Balanced vital coenzymes such as heme and minerals also must have contributed for the promotion effect on many cultures. Therefore, one might call this single-cell protein ingredient as an excellent alternative protein, which could be an ideal ingredient for MediFood or care food formulation in the future. I close my talk with appreciation to my colleagues and the Korean ministries for funding and regulation advice. Many thanks for the presentation opportunity and for your kind attention.